From Microbe TV, this is Immune, episode number 81, recorded on May 5th, 2024.
Hi, everyone. Welcome to Immune. We are at the American Association of Immunologists 2024 conference in the Windy City of Chicago. We are here interviewing excellent scientists about their work and their labs, and we have a guest here today that I will introduce to you in a second. But first, I'm Stephanie Langle, and here with me today is Brianne Barker.
Hello.
And we are the podcast, The Body's Defenders Against Disease. And we want to say if you'd like to contribute to help support the type of content that you'll be watching today, you can go to microbe.tv/contribute, as we are a 501c3, so your contributions are tax deductible. And today I'm going to introduce Dr. Joseph Larkin from the University of Florida. He is an associate professor and is going to talk to us today about his research program. And I think we're going to kick it off by first asking you, when did you first fall in love with science? Was it early for you? Was it later? Tell us about that.
So that's a good question. So growing up, I was very fascinated about how things work. But my story and my love of science kind of is a little bit on the sad side because when I was about 9 years old, I wanted to watch television. So far, it's not really sad, right? But I turned on the television, and Saturday morning, you get your cereal, and you get in front of the TV, and we didn't have cable, so we had 3 channels: ABC, CBS. We had PBS too, but I'm not watching PBS on a Saturday, right? But I wanted to watch. I don't even know what I wanted to watch, but the St. Jude Telethon was on, and I'm like, oh, this is awful. I can't watch my cartoons. But then I realized as I was watching that if people don't help these kids are going to die. And it was at that moment I really developed a passion and wanting to help kids. And now, of course, I don't do that now, because I don't do, oncology. But that was really my interest in science.
Interesting. And so did that follow you in even through school? And how did you, did you know you could be a scientist? Was that something that was in your worldview, or how did you kind of formally then start your education?
Oh, so not at all. Okay. Not at all. So I, for a long time, I was pre-med because I thought that was the only thing that I could do. And I began to just do some work at school, and I had an amazing chemistry professor that I'm still on Facebook with. So
awesome.
And he really opened up the world of science to me, and it was really nice. So, but I still didn't understand the whole idea of doing research until probably when I started doing undergraduate research.
And where were these places at for you? Where was your undergrad? And I guess, where did you grow up? What state did you
grow up? So I was born and raised in Savannah, Georgia. And I assumed I was going to go to Emory University in Georgia. I never liked the Bulldogs, so that was things up. But my senior year in school, I was invited to come to the University of Florida to visit. And this is a kind of a huge deal for us because we were very modest in income. Modest is probably not the right word, but it was like a vacation for the family. And we went to the university, and I was a National Merit Scholar. So I was able to attend the University of Florida. So we went from, being in Savannah to Gainesville, Florida. And that's where we did our undergrad. And again, I'm at this point, very naive to the whole world of science. So I started doing undergraduate research with Howard Johnson. And it's okay if I mention him?
Oh, yeah, absolutely.
And I started doing undergraduate research, and that's where I really developed a passion for research and science because I realized that, for that brief period of time before Howard started to hound me, I was the only person in the world that knew something. Right. Mm-hmm. And that was the thing. I was hooked at that point because it's like, I know something that no one else does.
That's awesome. What did you work on?
I worked on signal transduction. I worked on interferon gamma signaling and basically understanding how that signal, once it bound to the receptor, how the JAK stats were involved and how it got to the nucleus.
Cool. So then if you weren't totally sure about research, how did you kind of decide to go to graduate school, and where did you end up?
So I stayed on with Howard at the University of Florida, Dr. Johnson, and that's where I got my PhD. Okay. And I guess some things in life, you want to, like, I had this great story about why I wanted to do science. Part of me wanting to become a researcher as opposed to a medical doctor was because I love the lab. I love the science. But when I volunteered at the hospital, I was tearing around ice buckets to people, right? They called me the Iceman. No, I was not in love with being the Iceman. So it's like either Iceman or science, and I went with the science.
Right.
Sounds good. Yeah.
Oh, you like the story?
I do. I like it. So then you stayed on, still working on JAK-STAT signaling.
So I continued on with JAK-STAT signaling. Did some confocal microscopy. It was all the rage at the time. I guess it still is. It's still pretty cool. And so I focused on the Janus kinases, looking at localization of stats going to the nucleus and looking at some of the corresponding activation events.
That's great. So you're in graduate school at the University of Florida, and you're thinking at that point, do you know what direction you want to go in terms of a career? Can you tell us about that?
Yeah. So I guess I was a little bit winging it at the time, right? So at some point, and I did reasonably well in graduate school, but again, because it was so new to me, I had no idea what to compare it to, right? So, but I was doing well, and so I started looking at postdocs. And I had this grand scheme. Let's go to a university that I know, right? That I've heard before. So I knew of work that was going on at University of Pittsburgh. I knew of some of the things going on at Johns Hopkins, and then also I'd heard of the Ivy League and the University of Pennsylvania. So I applied to those places, and I received interviews at all three. But then I went to University of Pennsylvania. I met Yvonne Patterson, who is just an amazing, amazing person. And I went to University of Pennsylvania, and I was accepted on a T32 training grant. And then I started working in the lab of Dr. Andrew Caton there on that training grant. So that's
awesome.
What made that.
And so then what did you work on there?
And so I changed a little bit. And so it's funny because growing up and you tell, and people tell, well, your parents tell people that you are a doctor, right? And that means that so you know nothing, you become a doctor. Now you know everything, right? But I didn't know everything when I went and I started my postdoc. It was actually a rather steep learning curve because I went from signal transduction, largely cell lines, in vitro systems. We did a little bit of antibody production with rabbits, but most of it was an in vitro system to a system where Andy has this huge room of transgenic mice. So it was quite the different thing.
Yeah.
But it was an exciting time, and I was able to interact with, and I'll talk about what I was doing, but I was able to interact with Chris Hunter there, and Yvonne was there, and there was just I think it was at the time there were 70 immunologists, so it was just overwhelming, but in a really good way, where you could just soak in so much information. But in Andy's lab, there were these things that there was a rumor that there was this population of cells known as regulatory T cells, because at the time, they didn't really exist, right?
Mm-hmm.
So this is about 2000. And so I went to the lab, and I was really interested in, interestingly enough, the role of regulatory T cells in maternal. Yeah.
Okay.
But honestly, I never actually worked on that, right? But that was why I started the role of regulatory T cells in the maternal baby interface, right? So everything kind of goes
it does
full
go
circle. But what I focused on really was thymic development of this population of cells that may or may not really exist, right? So we looked at thymic development, but also in these animal model systems, and I work with Andy Rankin, Heath Gay, just amazing graduate students in the lab. In one system, because of this, let me explain the system just a little bit. So you have a TCR transgenic mouse, and then you have a mouse that expresses influenza as a neoantigen or self-antigen. So when you mate them, the progeny are expressing the HA, but there are also these T cells that are specific to the HA. So then as would be expected, a lot of it was thymic deletion, right? But in two strains of mouse, there was and I haven't talked about this in a while.
Bringing it back.
There were instead of the massive thymic deletion, you actually had comparable numbers as the TCR transgenic by itself. So, but when you look at those populations in the TS1 by 28 and the TS1 by PEV, and it's just how the antigen was being expressed in the different systems, half of the cells were CD4 positive, 25 positive, and the other half were CD4 positive, 25 negative. And so based on the markers of 25 positivity and 25 negativity, because FOXP3 wasn't known at the time because Rudinsky and his group hadn't done it until about 3 years later, we could identify antigen-specific regulatory T cells, which was a really cool system to work on. So in addition to looking at some of the thymic developed events, and Martha Jordan, who is still at Penn, she was doing some work on this, a lot of the work, leading work on this. We continued on some of this thymic development, but also looking at the activation of those regulatory T cells. And one of the really cool things that we were able to show is that we oftentimes think of T cells just kind of doing something. Okay, now you go proliferate, right? You go produce cytokines, right? But they're doing a lot of things, right? And so I can look at the proliferation of the 25 negatives while looking at the suppressive capacity of the 25 positives. And what I was able to show is that in cases where you just got a little bit of stimulation, whereas you would get no proliferation, you could still get 100% suppression, right? And that was something that I thought was really interesting and exciting.
Yeah. And so that experience, did that shape how you saw yourself as a scientist moving forward? Was this the field that you would want to start your own lab with? Or were you still at that point thinking, do I want to have my own lab? What were your thoughts back at that time? So
I was still a little bit the little engine that could, right? I think I can, I think I can, I think I can, I think I can. And I don't think about at that point, even though I didn't doubt my ability to generate data, and even though I was leading things in different ways, I didn't necessarily see myself as the leader of a lab, right? And I'm not saying that I didn't see it, but it wasn't this concrete thing in my mind at that point. So I continued on, because what do you do in science? You become a medical doctor, you work on. And that's what I had in my mind. So I don't know if I really, even at that point, envisioned a career as an academic professor, right? But it was leaning towards that way, right? It began. So we continued on working at UPenn, And then we were, I talked to Howard Johnson, my mentor at UF. And he says, "Oh, we have an opening here." I mean, it was about that time because I'm looking,
Sure.
and maybe a little whining, like, what am I going to do, and
that's usually part of a postdoc.
Oh no, that's definitely part of a postdoc.
Yeah.
And I was invited back. I guess
Okay. Oh, that's great. That's talking about full circle stories. I mean, that's really exciting to be able to go back to the place where you spent so much time and you had a deep history with, both scientifically and personally.
Yeah, and clearly they liked you.
Yeah, clearly.
So
then what did you start your lab working on?
So then we, so at the Wistar, we did regulatory T cells. But in one of Andy's expression systems, you got massive deletion of so many cells from the thymus. And also some of the regulatory T cells also got deleted. So it turned into a rheumatoid arthritis-like phenotype, right? Which is still really interesting.
Yeah.
But then Dr. Jan Erickson was working on MRL, LPR, GLD. What's that? Well, these are mice that go on to develop a lupus-like disease. And so, and as you say, how are you being shaped, right? Sometimes you're being shaped in ways you don't even really realize, right? So, so we're doing signal transduction. So then we started focusing on cellular immunology in these mouse models, right? And so that's how, the regulatory T cells are then interacting with the conventional T cells, and now the T cells are shaping the antibody responses. So you're just getting all this and you're soaking it all in. And now, based on how these cells interact with one another, they can influence whether or not a mouse, because at the time I was 100% mouse doctor, right? I cured a mouse. How these interactions can then go on to determine autoimmunity. So then having these kind of thoughts bouncing around in my head, I go back and Howard is working on, SOX, and I've heard of it, but I haven't really focused on it. And so the SOX are suppressors of cytokine signaling, and they inhibit the cytokine responses. So signal transduction. So now coming back, I'm back into the signal transduction field, but as opposed to looking at how the signal is being propagated to the nucleus, we're looking at how we can turn it off, right? Which is really important in terms of any immune response, right? You really want it to have, even that might even be more important, to be able to turn it off as opposed to it starting. So we started working on suppressor cytokine signaling. We were thinking about our regulatory T cells. We were thinking about autoimmunity. So then we combined things together and so then we started looking at how suppressors of cytokine signaling could potentially influence the regulatory T cell population. And the way that we initially started to work on that is that we, Jim Eiley had some mice that were SOX knockout of St. Jude, and we were able to get some of those mice. And so SOX1 is so important that if you have it completely knocked out, mice die within 21 days. And so we obtained 2 heterozygous pairs to start our breeding colony. And that was really exciting, right? Because that was kind of my first thing, right? And you're just staring at mice, dude, you've seen mice before. Andy had this huge colony, but these were my mice.
Your
right?
mice.
Right.
And so if you have a FOXP3 deficiency, then people suffer a really bad disease, and then of course the mice, the scurfy mice, they die within a similar period, right? So our idea was wonder if this, the regulatory T cells and the SOX1 were related, and that's where we we kind of began our studies looking at that. And then also in the literature, when you talk about these animals that are deficient in SOX1, one of the things that they tended to develop was a disease that looked a lot like lupus, right? So we started working on lupus because Jan had these mice, but now we began to look at regulatory T cells, SOX1, and autoimmunity. And that was the focus of the lab, and it still is right now. And we've expanded that. I don't know if you want me to talk.
Yeah, I would love to.
As I say, that is one of the ways that we got connected, because during COVID, the SARS-CoV-2 outbreak, you also expanded in some interesting breast milk related research.
Yes,
you had a maternal interest back with T regulatory cells. So
I did. Yes,
it does come again full circle.
It does come full circle. Yeah.
But you can talk about those expansions, and then also how you think about your lab in the next 5 years.
Oh, well, that's very cool. And we're okay with time?
Yeah. Oh yeah.
Okay,
we're good.
Awesome. Yeah. So we started working on this, the regulatory T cells and SOX1, and it's kind of interesting because although regulatory T cells are known to, in some cases, they can be a sink for interleukin-2, to sequester it from our conventional cells. And, in some of our earlier work, we showed that it can inhibit IL-6 production. It can inhibit interferon gamma production. The regulatory T cell is actually still interacting and responding to these signals, which is kind of interesting. So now if you have this suppressor of cytokine signaling, now you have your regulatory T cell, and by influencing how it sees the environment, it can also possibly influence what it does. And so in these mice, we saw a really big reduction in the number of regulatory T cells. And so something that I failed to mention is that so Howard also had a mimic of suppressor cytokine signaling 1. And so this mimic, so SOX has different regions that are important, but two of the really important regions in terms of it regulating immune responses is that it has a SOX box. And so going back to our signal transduction, we have the plasma membrane, then we have the nucleus. And the way signal transduction, it works is that you have a signal at the plasma membrane, and it's like, how does it get to the nucleus, right? To tell it to do stuff, right? So SOX, what it does is, so you have all these proteins that are along the line that are helping to propagate the signal. So what SOX does is it ubiquitinates and targets these proteins to proteasomal degradation. So it's kind of like if you have a railroad track, then it pulls out some of the rails, right?
In the middle. And can't go any further.
And turns up the signal. So that's one way. And so the other way is that the kinase inhibitory region is kind of like a wrench that goes into the Janus kinase and prevents it from adding the phosphate groups to things, right? So things don't become phosphorylated, right? So it inhibits the phosphorylation of the stats, right? So you throw the wrench in there and it stops, right? So Howard's molecule is a lot like the wrench, right? So it throws it in there and it turns up the signal. So when we used the SOX mimetic in this mouse model where all the mice die within 18 days, so overall all the mice lived longer, but about 20% of them lived up to around 40 days, right? And whereas if you look at the SOX knockout, it's a little runty, really unhappy thing that doesn't eat, it really fails to thrive. We got a really clear phenotype where this mouse wasn't exactly wild type, but it wasn't the runty mouse. So at that moment, I said, hey, maybe I could be a scientist, a real bona fide academic researcher.
Wow.
Right?
Okay. Yeah.
So then we extended that, and I'll get into some breast milk. I know you're interested in talking about the breast milk, but we began to focus on the ability of this peptide that's kind of restoring SOX1, and there's been some literature, previous literature, and also some recent literature that suggests that either failures in the production of SOX1 or in its lack of stability is related to human disease, right? And so we believe that SOX1 is critically involved in regulating lupus. And we went back into the MRL LPR mouse model, and by peritoneally ejecting the SOX1 peptide, we reduced some autoantibody production, which is a hallmark of lupus. We improved some of the glomerulonephritis in these mice, and this is an ongoing thing that we want to go back into because we think we can do it better and stronger and faster. And around that same time, we started working with Dr. Charles Iguagu at the National Eye Institute. And one of the things that he studies is uveitis. And he also studies animal model multiple sclerosis, but he studies uveitis, and uveitis is inflammation of the uveal tract. And so you have, the uveal tract, which consists of the choroid, the ciliary body, well, it's in the eye. And I go, well, if peritoneal injection of the peptide works, what if we make an eye drop?
Oh.
And so we took this eye drop that's containing the SOX1 peptide, and it's like, like in football where people draw up the football play, and it's like, okay, this guy's going here, this going here, and then you can throw in them and throw the touchdown pass. It works so well, right? It just was amazing. And so we took this, and I'll get to breast milk, but we took this, and I was talking to a congressperson, and which is could be scary, right? But one of my students wanted to talk to our congressperson. And so, so we went to Washington, and we started talking, and the congressperson was being very congressperson-like, actually very nice. I didn't vote for him, but very nice. But then I go, and we're doing this work where we mitigate uveitis, and he stops in his tracks. He stops being political, and he's probably listening. But he stops. He goes, if you cure uveitis in horses, you've really done something. I go, wow, really? And I'm a mouse doctor, right? It stops with the mice, right? And he goes, yeah, because it's impossible to treat uveitis in horses. And I go, are you challenging me? So we started this collaboration, and so Karen Plummer is one of the best veterinary ophthalmologists in the world, but she just happens to be at UF. The nicest person, but I go in, I start talking to her. It's not going to work. Like you could just see it on her face. It's not going to work. But she's humoring me. She's like, well, we can try. So we started trying it. And of course, so we did this open label clinical trial. And it reduced pain for the horses. And it was being used as a monotherapy. And granted, these horses were at advanced stages of disease where these horses were now refractory to the classical steroid treatments. But it reduced the redness, it reduced the pain. And we had someone other than Karen look, and it really had these effects in horses, right? And if you look at the sequence of JAK2 in horses and in people, the region where the peptide or the drug works is exactly the same, right? So we really think that there's some translational ability there. But Lauren wanted none of that when she came to the lab, because at the time,
and Lauren's your PhD student.
And Lauren is my PhD student. Well, now my momentary postdoc, because she gets a raise. Dagnabbit. What's up with that?
Because the NIH just put out that. Is that what you're talking about?
She gets a raise. No, she's deserving that and more. But naturally, she's actually done some work in the uveitis space as well. But when Lauren first came to my lab as a, just receiving her master's degree. Shortly after she came to the University of Florida, she left. And I'm like, oh no, because there was a pandemic going on, right? That stopped the world. And it's like, are you coming back? Well, so she comes back, and I said, well, maybe you'll be interested in, doing something where you can be involved in COVID, right? And she's like, I'm listening. And so I said, I know someone at the university, Dr. Joe New. He has a fellow that is interested in breast milk and also interested in whether or not people that are vaccinated, whether or not they have the ability to generate SARS-CoV specific antibodies in their breast milk, and two, if they can pass them on to their baby. And so she's like, I'm listening, and and she comes in, and Lauren's she's just amazing. You're going to be happy.
I'm excited. We'll break the fourth wall to say that she is coming to my lab to be a postdoc in August. So
she's fantastic. And it's almost surprising just for you, because I have this idea, and she's listening, and I come back and I go, well, about that idea, and she goes, oh yeah, I ran an experiment, and yes.
Oh, you love that.
Yes.
Really?
Like, oh, you've already ran it.
We've already run this experiment that I didn't even fully formulate in my mind yet. But she, fantastic. So we were recruiting, and so it's really kind of an exciting time because, one, we've got mRNA vaccines, right? What is that, right? So we're learning about the mRNA vaccines. We have this virus that stopped the world, right? People are becoming vaccinated. And who gets vaccinated first? Well, people that are, involved, they're in the hospitals. I was working, with the SARS-CoV, and I couldn't get vaccinated, right? But, but other people could. That wasn't right. But we have the people that are being vaccinated at the hospital. Vivian was one of the people that was vaccinated, and she was also, I guess she was nursing. Can I say that she was part of this? She was a participant in our.
I don't know. But
is that a sort of violation? Can you cut that part? I don't know. Right. Well, we have people that are being vaccinated, and so we saw people that had no vaccination, the first dose, the second dose, and then we also had people that were taking the booster, and so we have this cohort, and so we see that with the first vaccination, we see antibodies present within their blood, which is no big surprise because that's kind of been published. But we see the antibodies in the breast milk, and they were the IgAs largely. With the second vaccination, there was a huge jump in the amount of IgAs that were SARS-CoV specific. And then I'll hold off on the booster. But then we also looked in the babies because, moms are very squeamish about having their babies bled, right? But they love to give you poopy diapers. They had no problem giving us. Would you like some more? No, we're good. We're good. So we were able to see the antibodies present in the stool of the babies, and that's kind of exciting. So so of course we were never asking the question of what degree of protection was being conveyed, but we clearly had the smoking gun that it was being transferred. So that was very exciting, and so we were able to run the experiment, and then we looked at the booster. And I thought that it would be a lot more IgAs. And so it's like, this is going to be good, but I already know what's going to happen. But it turned out that there was an isotype switch there, right? Which I think is really an important future direction, because the SARS-CoV specific antibodies present within the breast milk were largely IgGs. And that was not what I would have
Yeah, me neither.
Right? So we started looking in the literature, and there is evidence that IgG-specific antibodies in breast milk have had protection in different viral responses. So I really think that there's something there, because the first thing that came to my mind is like, oh, well, nature kind of messed up on this one. No. So I'm very, and so in the literature, there are so it's well established that the neonatal receptor is important for if the baby's in utero. Then mom can transfer her IgGs to the baby through the neonatal FC receptor. But there is I wouldn't say it's overflowing, but there's evidence that there are also neonatal FC receptors present within the lumen.
Mm-hmm.
Okay.
And so as a future, I'd like to see what those IgGs are actually doing.
Sure.
In the lumen, because in, as you know, in many species, when the baby is not fully developed, the antibodies can just kind of pass through because the tight junctions are not so tight in the gut. But in people, it's pretty tight, right? Yeah. So I think that maybe there's some active transport going on there. So that's something that we're really interested in. We're trying we really do believe that SOX1 eye drops are going to work. Mm-hmm. So we're interested in that, and we'd like to go back into lupus and make some headway in there.
Wonderful.
Yeah.
Well, I think, this has been really great to hear your story and all the great work you're doing. And I guess as one last question, you're on the AAI Diversity Committee, and would be curious to hear if you have any thoughts about that and your experience with that committee.
Okay. So I was on the committee. I've rotated off, but it's a great committee, and there are people that have become lifelong friends. And I've graduated 9 PhD students. I've chaired 9 committees, and 7 of the 9 were female, and all of them were either of underrepresented or economically disadvantaged. Right. So I am very passionate about promoting diversity in terms of research, and sure, I mean, one could think about it being a selfish thing, right? And yeah, I mean, it is a little bit selfish, but diversity, and even in in nature, if we have a gut that's just full of one bacteria, we're not getting everything that we need, right? So different people from different backgrounds, all 3 of us can look at some car getting a crash on the street, right? But whether we like it or not, how we see that is going to be influenced by the lens by which we have seen life.
Of course.
So diversity brings in a different lenses, and it just makes things better. So being a part of that great organization, and being a part of AAI, who is invested in promoting diversity, is something that is very near and dear to me.
That's awesome. I feel like between all of the work that you're doing with eye drops and with lupus and with breast milk, and even thinking about diversity in science, you're helping a lot of those kids that you were thinking about at the very beginning.
From St.
Jude.
Yeah.
Thank you. Yeah. I think we're doing our part. I actually yesterday I was at the table, the mentoring session, and a young man starts coming up to me. He's got this big smile on his face, and I go, "Oh, Jamal, I know you." It turns out that he was one of my undergrad that takes my undergrad immunology course.
Oh, that's cool.
And he was still smiling, right? Everyone that takes my classes not smiling afterwards. Just looking at my professors, they're not smiling. But he just defended his PhD.
Oh, wow.
Awesome.
And now he's MD, PhD at Penn. So I guess he's going to go back into the clinics and do his MD part. But that really felt like I had done something because this young man, he's doing quite well.
Awesome.
That's great.
That is fantastic. Well, that brings us to an end of a very special episode of Immune. If you have questions or comments, you can email them to immune@microbe.tv. Please consider supporting the science shows of Microbe TV at microbe.tv/contribute. Microbe TV is a 501c3, so your contributions are tax deductible. Joseph Larkin is at the University of Florida.
Go Gators.
Stephanie Langle is at Case Western Reserve University.
Yes, thank you. Thank you, Joe, for coming on.
Oh, this was great. Thank you.
Absolutely. Yeah.
I'm Brianne Barker from Drew University, and I'm so excited to have learned a lot today. We can be found at microbe.tv with a new episode on the last Tuesday of every month. Music on Immune is by Steve Neal. Thank you for listening to Immune, the podcast that's infectious. We'll be back next month.